Elevated cholesterol is a major risk factor for cardiovascular and neurodegenerative disease. Our protocols provide valuable tools for studying both physiological and mechanistic consequences of hypercholesterolemia.
These procedures can be performed using basic lab equipment and are applicable to cells, tissues, and Xenopus oocytes. Cholesterol is a major component of cellular membranes throughout the body. These techniques can be utilized to study the impact of elevated levels of cholesterol in any cell type.
Dissecting the arteries is the most difficult step. Carefully remove the artery from the brain without stretching or damaging the vascular tissue.
Lipids are very delicate, and working with them is more of an art than a science. Video demonstration provides a guide for learning how to handle lipids carefully.
To prepare the cholesterol-saturated methyl-beta cyclodextrin solution, first add 0.064 grams of methyl-beta cyclodextrin to 10 milliliters of PBS, stirring the solution with a stir bar to ensure that the methyl-beta cyclodextrin fully dissolves. Next, add 0.0024 grams of cholesterol powder to the flask, and stir the solution vigorously, using a spatula to break up as many cholesterol chunks as possible. When nearly all the cholesterol has been dissolved, seal the flask with at least 2 layers of paraffin film. And shake the flask at about 30 oscillations per minute in a 37 degrees Celsius water bath overnight. After 8 to 16 hours, cool the solution to room temperature.